Hello students, in this part we will discuss on oogenesis, formation of female gamete ovum. First one is oogenesis. What is oogenesis? Formation of female gamete ovum or egg, that is called oogenesis. We have already discussed spermatogenesis is a continuous process, because if one time the production of sperm may start, that remains continuous. But oogenesis process is a discontinuous process. It is discontinuous process. First of all, we will study how these eggs are produced inside the female ovary. Gestation period, the period through that the time period which is taken by the baby from after fertilization to delivery, that time period is called gestation period. When the female fetus remains inside the body of the mother, at the end of first month, there are 1700 primary follicles are produced in the female ovary. First month of the female fetus, there are 1700 primary follicles or follicles are produced inside the female baby in the ovary. But this breakdown or the decomposition, breakdown in the primary follicles, continuous process can take place, and the end of the second month of the embryo, the number of primary follicles increase due to the division, and it increased from 1700 to 6 lakhs. Thus, the division in primary follicles continuous can take place, and at the end of 5th month of the embryo, the primary follicles numbers are increased to 7 millions. 7 millions. After the 5th month, these primary follicles start to degenerate. Degeneration of primary follicle, that process is called follicular atresia. It means after the 5th month of gestation period, the degeneration of follicles. The process in which degeneration of follicles can take place, that is called follicular atresia. What is follicular atresia? Degeneration of follicles. These follicles after degeneration remains at the birth time in the female is nearly 2 millions. At the time of baby birth, these primary follicles remains only 2 millions. Then these 2 million primary follicles at present remain same and continue start to degeneration as the female reach to the puberty. These primary follicles only remains at the time of puberty in females 60,000 to 80,000. It means we can say that at the time of puberty, in female, the primary follicle the number of primary follicles remains 60,000 to 80,000. As we have discussed, oogenesis is a discontinuous process. How this discontinuous process can take place? We have studied that the first month, second and fifth month, the number of primary follicles increases. But after the fifth month, due to follicular atresia, the number of primary follicles reduced. So we can say that oogenesis process is a discontinuous process, whereas spermatogenesis in male is a continuous process. First of all, this one. And second one is how is the oogenesis process can take place. The oogenesis process completed in 3 phases. The oogenesis process is completed in 3 phases, and those 3 phases are: first is multiplication phase, second one is growth phase, and third one is maturation phase. Just like as spermatogenesis, there are 3 phases are also occurred in oogenesis. One more thing which we have forgotten: at the time of puberty, there are 60,000 to 80,000 primary follicles present. Every month after the puberty, every month ovary release an egg. So we can say that out of these 60 to 80,000 primary follicles, nearly 500 primary follicles out of 500 primary follicles, only one primary follicle develops, and the rest, the primary follicle produce around another layer to the primary follicle which is developed. It means nearly 500 primary follicles. Out of 500 primary follicles, only one primary follicle produce or development can take place. The rest primary follicles make a layer around this developed primary follicles, that is called Graafian follicle. Clear? Then after that we have studied that oogenesis process is completed in 3 phases, and those 3 phases are multiplication phase, growth phase, and maturation phase. First of all, we discuss how this oogenesis process can take place. We have studied the first phase is multiplication phase. Multiplication phase. The development of follicles from the ovarian epidermis layer. Thus, this is the ovarian epidermis layer. The primary follicles are produced from this one. Primary follicles are produced from this one. And this primary follicle, the primary follicles, which develops from the epidermis of ovarian wall, which is a diploid cell, Just this cell, which is produced from ovarian wall, this cell is a diploid cell. This diploid cell is known as oogonia. Because due to the division in the ovarian wall, the primary follicles are produced, and these cells increase their numbers. So this is called multiplication phase, and here is the division can take place, mitotic division. So we can see that due to mitotic division in ovarian wall, that produce the ovum or oocyte primary follicles. These are called oogonia. Number of cell increases. Number of cells increases. How? Because due to the mitotic division. This mitotic division can take place in ovarian wall. So the division in ovarian wall produce the follicles, and these follicles are known as oogonia, and these are diploid cells. This is called multiplication phase. After the multiplication phase, the cell start to develop its size, and that phase is known as growth phase. This is the multiplication phase because they increase their number. The first phase. Then the cell start to grow its size, and that is called growth phase. How the cell increase their size? There is the formation of yolk can take place, and the formation of yolk, that process known as vitellogenesis. So we can say that vitellogenesis process. What is vitellogenesis process? Formation of yolk. Formation of yolk in the egg, that process known as vitellogenesis. So the oogonia, due to the formation of yolk, start to increase its size, and that increase in size, which is a result of the size increases in the cell, and this is cell size increases, and this is called primary oocyte. Primary oocyte. As the yolk is formed inside the oogonia, first of all, that process is known as vitellogenesis, and phase, due to the formation of yolk, the cell size increase, so that phase is known as growth phase. Due to the formation of yolk, another thing, how the cell size increases, what factors affect the increases in the cells? We have studied that there are two types of organism. First one is viviparous organism, and second one is oviparous organism. Organisms are, or animals are, we consider two types: oviparous and viviparous. First of all, what do you understand by oviparous animals? Those animals which lay eggs. Those are called oviparous organisms, mostly birds. And the animals which produce young ones, those are called viviparous, for example, human. The formation of yolk quantity is dependent on these two types. If the organisms are viviparous, if the animals are viviparous, so we can say that development of baby can take place inside the mother body. And inside the mother body, the developing embryo take the nutrition through placenta. So there is less quantity of yolk formation can take place. Less quantity of yolk formation can take place. The size of the egg is also increases in less quantity. But the animals which lay eggs, it means they, after the laying eggs, the embryo development can take place. So there is no placenta produced. In these type of animals or these types of eggs, formation of yolk in excess quantity, and due to formation of in excess quantity, the cell size or egg size increases. How the egg size increases? In human being, in human being or human female, the egg size increases from normal to 200 times more. In hens, egg size also increases in 200 times. In frogs lay eggs. After they lay eggs, fertilization also can take place outside the body. So the yolk formation can take place more quantity. In frog, the egg size increases 2000 times from their normal. And in Drosophila melanogaster, is a fruit fly. So we can say that in fruit fly, the cell size increased from normal to 90,000 times more. This size increases dependent on the required nutrition. If the embryo remains inside the mother body, the nutrition provided through the placenta. So there is no formation of yolk in excess quantity can take place. But if the cell eggs are released through outside the body, development of embryo can take place outside the body. These type of cell require excess quantity of yolk. So the amount of yolk formation is required is excess. Therefore, in frogs and Drosophila melanogaster, the cell size, the egg size increases the maximum time. Clear? After gaining the formation of yolk, the cell size increases, and this is called growth phase. Then this growth cell, primary oocyte, enters into the another phase, and that phase is meiosis 1. Means meiosis division can take place. After this, the meiosis division can take place. Meiosis division, so cell increases its number, but in female is an exceptional case. Human female is an exceptional case. Due to the meiosis division, one primary oocyte can produce only one secondary oocyte, not two, because the most cytoplasm yolk present in it, breakdown can take place and it produce a single cell, a small cell, and one another cell which contain excess quantity of cytoplasm. Therefore, this will happen in M1 phase. M1 phase, meiosis 1 phase. Meiosis 1 phase is a reductional phase. Here is primary oocyte is a diploid cell, because mitotic division can take place, no other division can take place. Mitotic division is equational. So, ovarian ward to oogonia, diploid to oogonia to develop primary oocyte is also developed. A diploid cell, two set of chromosomes. As the primary oocyte cell increases its size, it enters into the meiosis 1. And meiosis 1 is a reductional phase. Reductional phase. So the two set of chromosomes becomes half, but the cytoplasm in one cell comes in very less quantity, and another contains excess. This cell is a haploid cell, and this one is also haploid, because meiosis division can take place. Two set of chromosomes divided remains half. One set of chromosome remains in this one, and one set of chromosome remain in this cell. The cell, this is called secondary oocyte. This is called secondary oocyte. It is a haploid cell. And the other tiny cell, this is called first polar body. After some time later, the polar body degenerates, only single secondary cells remains. Secondary cell, this is the cell when the egg is released through the ovary, that is called ovulation. If there is no fertilization can take place, secondary oocyte is released. This is released through ovary ovulation. Clear? After the realization, after the ovulation, if this cell got sperm, if the sperm triggered this cell, entry the sperm of this cell, another division can take place, that is called meiosis 2. If suppose this cell gain sperm due to triggers by sperm, or we can say that due to trigger entry of sperm into that, then another division can take place. Another division, meiosis 1. Meiosis 1, then this cell is called ooted egg ovum. This is called ooted egg or ovum. Another tiny cell is also produced. This tiny cell is called second polar body. Second polar body. Second polar body can take place only in one situation, if the the sperm is entered into the egg. Otherwise, this is released in the form of secondary oocyte. Then this ooted means egg, fertilized egg, moves through the fallopian tubes by the peristalsis movement and reach to the uterus and implant at the inner wall of uterus and start to development of embryo will take place. This is the division in the ovary, or you can say that ovum can take place. First of all, we listen that oogenesis process is completed in 3 phases. First phase is multiplication phase. It means the cell size, cell number increases. How the cell increases? Ovarian wall start to division. Which division? Mitotic division. Ovarian wall contains diploid cell. The mitotic division is same. So that cell produced from the ovarian wall is diploid, which is known as oogonia. This is called multiplication phase. After the multiplication phase, the formation of yolk inside the cell, inside the oogonia, can take place, and that process is known as vitellogenesis. What is the amount of yolk can produce inside the cell, which is dependent on the requirement of nutrition. If the animals are viviparous, less quantity of yolk formation can take place because the mostly nutritional substances are provided by the placenta. But if the animals are oviparous, these animals lay eggs. It means the development of embryo can take place outside the body. So the nutritional substances are required in excess quantity. Due to formation of excess quantity and due to requirement of excess quantity of nutrition, yolk is produced excess. So we can say that how the size of cell increases. How many times? In human female 200 times. Drosophila melanogaster 90,000 times more, frog 2,000 times more. This will happen due to the requirement of nutritious substances. And the cell size increases. That phase is known as growth phase. After gaining the growth, the oogonia changes to primary follicle. That is also a diploid cell because there is no division can take place. Diploid cell. Then this growth primary oocyte enters into the meiosis 1 phase. Meiosis 1 phase is a reductional phase. One reductional phase, which is exceptional case in human female, that is 2 cells are produced. One is smaller and other one is larger. The larger cell contains mostly number of cytoplasm, excess quantity of cytoplasm, but a tiny cell, it contains one set of chromosome, but very few or negligible quantity of cytoplasm. It contains large quantity of cytoplasm. The tiny cell which is produced, which contains a negligible amount of cytoplasm, that is called polar body. This is the first division, so it is called first polar body. After this one, the first polar body degenerated. This is called secondary oocyte. At this age, ovary release this secondary oocyte. After that, if this cell got sperm, if this cell means secondary oocyte cell got sperm, the fused cell is known as egg. And then meiosis 2, M2 phase can be occurred here. M2 phase, in M2 phase, one another tiny cell produced. That is the second situation. So that is called second polar body. And one another large cell which contain excess quantity of cytoplasm. This is called ooted egg or ovum. Because this is fertilized egg. And after gaining the fertilization, this fertilized egg move through the fallopian tube with the help of cilia, that process known as peristalsis movement, and enters into uterus. At the uterus inner wall endometrium, this fertilized egg are staked. This is called implantation can take place, and this whole process is known as oogenesis. In next part, we will discuss on structure of oogenesis as well as menstruation cycle in female.